Welcome. In this video, we talk about conjugation, which is a process of gene exchange in bacteria. Now, it is important to know that bacterial exchange through genes occurs in three ways, that is through conjugation, transformation and transduction. These are the three ways in which bacteria can exchange gene. Now, bacterial exchange genetic material by three different mechanisms. Right, so the first one is the conjugation, which takes place when genetic material passes directly from one bacterium to another. In conjugation, two bacteria lie close together and connection forms between them. So if this is a bacteria, it forms a pili and it connects with another bacteria. This should be F plus and F minus. We talk about what they are, and then its genetic material can be transferred. So this is conjugation, where the two bacteria lie close together and a connection between them is formed. This is a connection known as pili forms between them, and a plasmid or a part of the bacterial chromosome passes from one cell, the donor, to the recipient. So because the DNA here is passing from this one, so this is donor and this is the recipient. Subsequent to conjugation, crossing over may take place between two homologous sequences in the transferred DNA. So after the transfer of DNA had occurred, here crossing over between the two DNA can occur. In conjugation, DNA is transferred only from donor to recipient with no reciprocal exchange of genetic material. But it is important to note that in conjugation, only transfer of DNA only occur from the donor to the recipient, that is from here to here. No reciprocal transfer occurs. This does not happen. Second phase, of course, transformation, which takes place when bacterium takes up DNA from medium in which it is growing. So if it is a bacteria, so it takes up DNA from the surrounding material, right, and then crossing over takes place between them. After transformation, recombination may take place between the introduced genes and those of the bacterial chromosome, right, while transduction takes place when bacterial viruses, bacteriophage, carry DNA from one bacterium to the other. It is a complex mechanism. We will study about it in the next video. This video, on the other hand, focuses only on the conjugation. Here is the picture showing conjugation, which has been taken from Benjamin and Pierre's book. You can see it. So conjugation, we can see that this is the donor cell, this is the recipient cell, and both of them have their bacterial chromosome, this one and this one. So what happens is that a cytoplasmic bridge called pili forms, right? A cytoplasmic bridge forms between the two bacteria. Now DNA replicates and transferred from one cell to the other. So here we see that the DNA is transferred. Here it goes from this donor to the recipient, and then transferred DNA replicates. As you can see here, it replicates. Now after replication, a crossover in the recipient cell occurs, right? So crossing over between the recipient DNA and the newly acquired DNA occurs, and the creation of a recombinant chromosome occurs, right? So it is a recombinant DNA is formed. This is how conjugation in bacteria occurs, right? So the next is, what is F plus and F minus cells? For studying conjugation, we need to know these things. So in most bacteria, conjugation depends on fertility factor that is present in a donor cell. So if conjugation occurs, it is important to know that the donor cell must always have its own DNA or chromosome plus a F factor. F factor must be there, which is also known as fertility factor. So along with its DNA, bacterial chromosome, it must also have a F factor to become a F plus cell to become a donor. Recipient cell doesn't need this F factor, but the donor cell does requires to be F plus or have a fertility factor. and absent in the recipient cell, while this F factor is absent in the recipient cell. Cells that contain F are referred as F plus and cell lacking F are F minus. So if this is a cell and this is a chromosomal DNA but it does not have a F factor, while this one has a chromosomal DNA plus an F factor, then this is F plus, a donor, and this is a F minus because it does not have a fertility factor. The F factor contains an origin of replication and a number of genes required for conjugation. For example, some of these genes encode the sex pili. So this F factor or the fertility factor is required, is very essential for the cell for the conjugation to occur. This is because this F factor contains many important genes like the formation of sex pili. Sex pili is the connection that forms between two cells, so it is required. A cell containing F produces the sex pili, one of which makes contact with the receptor on F minus cell and pulls the two cells together. DNA is then transferred from F plus cell to the F minus cell. Conjugation can take place only between a cell that possesses F and a cell that lacks F. So, and again I repeat, conjugation can only occur between a donor cell which is F plus and a recipient cell which must be F minus. So here we see the picture. So this is the F plus donor bacterium. Here it has the bacterial chromosome and it has the F factor 2. So it is a F plus cell and thus a donor bacterium, while this is a F minus recipient bacterium because it does not have a F factor. So here we see during conjugation a cytoplasmic connection forms between the two cells, that is sex pili is formed between the F plus and F minus cell. Then one DNA strand of the F factor is nicked at an origin and separates. So here from ORIT, an origin site at F factor, ORIT, origin site, the DNA is nicked and it starts to move in the F minus cell through the sex pili. Now the replication takes place on the F factor, replacing the nicked strand. After it has been transferred here, the replication had started. After the single strand replicates, producing a circular double-stranded copy of the F plasmid. So, if whole of the plasmid has been transferred here and it then replicates, so this F minus cell, the recipient cell that have been F minus till here, F minus, which did not have a F factor at the starting, has now a F factor and thus it has become a F plus cell. That is, it also have become a donor cell. At first it has been a recipient bacterium, but now it is a F plus donor bacterium because the whole of the F factor has been transferred into this cell. Now, in most cases, the only gene transferred during conjugation between F plus and F minus cells are those on the F factor. Transfer is initiated when one of the DNA strand on the F factor is nicked at an origin, that is ORIT. One end of the nicked DNA separates from the circle and passes into the recipient cell. The replication takes place on the nicked strand, proceeding around the circular plasmid in the F plus cell and replacing the transferred strand. As we saw in this picture, the F plus donor bacterium, F minus recipient bacterium, here is it has the F factor, and then from the ORIT, somewhere here, ORIT 5 prime, it starts to move here, like in this figure. So then it comes into the F minus cell, right, and then it starts to replicate in F minus cell and forms, so the F minus cell now becomes the F plus cell because the whole of the the F factor has been transferred. Now that we know what is a F plus cell and what is a F minus cell, we have to know about Hfr cells, that is high frequency cells, right? The conjugation transferred genetic material in F plasmid from F plus to F minus cells, but does not account for. So in Hfr or high frequency strains, what happens? The F factor is integrated into the bacterial chromosome. Here in this picture, as you can see, it is a F plus cell, and this is bacterial chromosome, and this is F factor. So what happens, crossing over takes place between the F factor and the chromosome. Here crossing over takes place between the bacterial chromosome and the F factor, and thus what we see is a Hfr cell has been formed, that the F factor is now integrated into the chromosome. So now the cell is an Hfr cell, which acts just like a F plus cell or as a donor cell. Right, so high frequency strains have the F factor integrated along with the bacterial chromosome. The Hfr cells behave just as F plus cells, forming sex pili and undergo conjugation with F minus cells. Now in conjugation between Hfr and F minus cells, the integrated F factor is nicked and the end of the nicked strand moves into the F minus cell, just like it happens in your F plus to F minus conjugation. In Hfr cells, the F factor has been integrated into the bacterial chromosome. The chromosome follows it into the recipient cell. How much of the bacterial chromosome is transferred depends on the length of the time that the two cells remain conjugation. Let's see what it means. So here we can see in this picture that this is a Hfr cell where the F factor has been integrated into the bacterial chromosome, and this is a F minus cell which does not have a fertility factor. Now in conjugation, the F is nicked at the 5' end and moves to the F minus cell. Here it starts moving into the F minus cell, and the transferred strand starts to replicate. This transferred strand starts to replicate. But along with the F factor here, the bacterial chromosome of this F plus bacteria or Hfr bacteria is also moving in. Right? You can see in the earlier case only the F factor was moving into the F minus cell. From F plus to F minus cell only the fertility factor was moving. But in case of Hfr cells, high frequency strains, because the F factor is integrated into the bacterial chromosome along with the F factor, the bacterial chromosome is also being transferred into this F minus bacteria. So here we can see Hfr chromosome, F factor plus bacterial genes have been transferred, and now crossing over takes place between the donated Hfr chromosome and the original chromosome of the F cell. Here along with this is bacterial chromosome, this is the F factor, and now crossing over between here and here takes place between the F minus bacterial chromosome and the newly acquired chromosome or DNA. Now after crossing over may lead to recombination of alleles, bright green in place of black segment. So here it had a black segment, so recombination had happened. Now it has the green allele after recombination, and the linear chromosome is degraded. Thus this F minus cell is here and recombination had occurred. Now after we know that what happens in Hfr cells, we also have to know about F dash cells. So in F dash cells, it is just opposite to Hfr cells. That is, for example, if this is a Hfr cell where the F factor is integrated into the chromosome, bacterial chromosome, crossing over takes place within the Hfr chromosome and the F factor excises from the bacterial chromosome. That is, it excises itself. It cuts itself out and again forms a separate F factor and a separate bacterial chromosome. Right? But there is a chance during this excision that it may carry some bacterial genes along with it. Like for example, here the lac gene was very close to this F factor, and after it is excised, the lac gene along with the F factor goes into the fertility factor or F factor, right, instead of the bacterial chromosome. So here F dash cell has the lac gene which is not generally present in F factor, right? So it is a F dash cell, and when conjugation between a F dash cell and a F minus cell occurs, there is a chance that along with the F factor, during conjugation, the F factor with the lac gene is transferred to the F minus cell, like here. So this is transferred, and the lag gene producing a partial diploid of 2 copies of lag gene. So along with the F factor, the lag gene is also being transferred in the F minus cell, this making it a F plus cell, or you can say F dash cell has been made. Thank you for watching. Please like and subscribe the channel.